thing stopped for some reason. Okay, let's start that again.
So when you have three different genes, unlike in dihypercrosses, where like the A's stood for one trait and the B's stood for another trait, in this case here, the A's, the B's, and the C's all determine skin color. What does that mean? It means that out of our 23 pairs of chromosomes, let's say in chromosome number 1, there's a skin color gene, and so this person is heterozygous, big A, little a. Then, let's say in chromosome pair number 11 out of our 23 pairs. So on chromosome pair number 11, there's another piece of part of the DNA that codes for making melanin and therefore codes for skin color. So the person's heterozygous for that gene. And then on, let's say, number 20, chromosome pair number 20, there's the big C and the little c. So you don't just have one skin color gene, you have multiple skin color genes and multiple pairs of chromosomes. And so what happens is that in this example, it's really much more than 6 genes, but in this example, there's 6 different genes. And out of the 6, this person has 3 out of those 6 dominant and 3 recessive, and it's the dominant ones that produce melanin. So every single dominant gene you have produces melanin. So therefore, they have just one dominant gene will produce a little bit of melanin. 2 dominant genes will produce more melanin. 3 dominant genes will produce even more melanin, and so on and so forth. And that's where the additive effect comes into play. So these 2 parents are both heterozygous for the skin color traits. This right here is the person's genotype for skin color. So you don't just have big R, little r for skin color, you have 3 different genes. And so you're big A, big B, little b, big C, little c for skin color. And so when these 2 individuals, which would have an intermediate skin color, why? Because out of the 6 genes, 3 of them code for producing melanin, so they'd be about the halfway point there. And so, so these guys, when they mate, they could have a kid that's just like themselves and be heterozygous. Notice that this one has 3 dominant genes, and on this chart here, so this is supposed to be the x-axis, so the different genotypes, and this is the frequency. And so, I'm looking at this. The majority have this intermediate genotype or phenotype, and so in this array of color, it would be intermediate skin color. These 2 parents could also have a kid that has 2 dominant genes. Now they gave an example that it's big A, big B, little c, and it has 2 dominant genes. A person that has a genotype like this, or like little a, little b, big C, these 2 individuals would have the same phenotype because they have the same number of dominant genes and therefore produce the same amount of melanin and therefore would have the same skin color. All right, so it doesn't have to be this particular genotype. And so then it's a little lighter skin, then 1 dominant gene lighter, all recessives the lightest. And then we can go in this direction here, so 4 dominant genes, a little bit darker than 3, 5 a little bit darker, 6 darkest. And so on. And so that's what it means by an additive effect. And this is called polygenic inheritance. Poly means many, and genic is referring to genes. So skin color is controlled by many pairs of genes. All right, and that's how we can get such a wide variety. If you had just dominant recessive, and there'd only be 3 different genotypes that somebody could be, and therefore only 3 different skin colors. All right, so having multiple gene pairs allows you a greater variety, and that's that continuum they were talking about. So in a population, we usually see when traits are inherited by polygenic inheritance, we usually see this bell-shaped curve where the more people are in the center, and then as you go to extremes, either extreme light or dark, or if it's height, extreme short versus tall people, the number of people go down in the population usually. And so whenever you see a trait and it's exhibited in this bell-shaped curve, which this shape is also called normal distribution. So if you were given information about a trait, that usually means that it's most likely a polygenic trait, or inherited by polygenic inheritance. creates that bell-shaped curve. And so you get this continuum there. So I need you to, that's one thing that you need to make that connection with, like as though if you see something with an unknown trait that you don't know about, if you see it where there's lots of different phenotypes and you see this bell-shaped curve in that distribution, it's most likely a polygenic trait. All right, and so so that's one other unique thing of how certain traits are inherited. The next thing that's unique is that some traits seem to be affected by the environment. And so sometimes I call this nature versus nurture, the environmental impact on phenotype. Nature is your genotype. And so they've done this looking at like twins separated at birth and grow up in two different families. So identical twins their genes are identical. So the genetics is identical. And then we look at how different environments can affect the overall traits of the organism, and therefore how much the environment affects certain genes and so on. And so that's the nurture part. So this departs from Mendelian genetics because he hadn't come up with necessarily that the environment is playing a role. So in the phenotype for a character, remember whenever you see the word character, it's trait. It depends on the environment as well as the genotype. And so this, what's called the norm of reaction, is the phenotypic range of a genotype influenced by the environment. So how much is the genotype influenced by the environment and change the phenotype? Let me give you an example of a situation that would be the case. Hydrangea flowers can be the same exact genotype, but express and have different color flowers, from this pretty bluish, purplish color to pink, depending on the environment. In this case, the environment is the soil acidity. And so what happens is, here's your two different hydrangea flowers. Their genotypes are identical. The one on the left is grown in soil that's acidic, the one on the right is not. So that acidic soil doesn't change the genotype. The genes are still the same, but the expression and what color that flower appears depends upon that acidic soil. So when you grow hydrangeas, you can actually buy in the store a substance that you can sprinkle and put into your soil and water it into your soil that makes your soil acidic, so it becomes that blue color. Because if your soil's not the default would be just the pink. All right, so that's an example of the environment changing or affecting the phenotype. Norms of reaction, or how the environment affects the phenotype, are broadest or affect the most with the polygenic characters or polygenic traits. And these traits are called multifactorial because both genetic and environmental factors collectively influence the phenotype. I'm going to talk more about multifactorial traits in humans in just a little bit, but I'll just address it a little bit right now. Two examples, heart disease and cancer would be an example of it. So heart disease has a genetic influence. So they say that if it runs in your family and you inherit the gene, you are genetically predisposed, which means that you have a higher likelihood of developing heart cancer, heart disease, or a certain type of cancer, let's say breast cancer runs in your family or something like that, than other people. But then the environment can also play a role. So like for heart disease, your environment, what you eat, how you exercise, and so on, can play a role in the manifestation of that heart disease in you. So you have both of those components going on. Same thing with cancer. So like, somebody can be genetically predisposed for a certain type of cancer, and then can influence that by putting things, let's say smoking, which is nicotine is a known carcinogenic, which could cause you to get that cancer maybe earlier in life than you would had you had a different environmental around you. Okay. So when we talk about a person or an organism's phenotype, it necessarily doesn't always have to be what we see, the physical appearance. A lot of genes control stuff that we can't see. So it can be behavior There's some genetic things with behavior. It can be what's going on inside, your anatomy, your physiology, what proteins you make and enzymes you make, and so on. And so the overall phenotype is influenced by the genotype and the environmental history of the person. All right? And so that's the last of the unique ways that things are inherited. This last section talks about how we trace and look at how different disorders and how different things are inherited in the population. Humans are not usually good subjects for genetic research. We take information about how things are passed down from generation to generation, but we don't do experiments with humans. First of all, we have a long generation time. You have 9 months to carry the baby, you have to have the baby has to grow up and have kids of their own to get data for the next generation, and so on. So there's big gaps between generations, and we don't have a lot of kids, so we don't get a lot of data, and so on. And then, as that last sentence says, breeding experiments are unacceptable. So we don't, like if you remember, I talked about test crosses. Like if you have an individual that's big R, or big R, little r, but you can't tell by looking at them, so you want to know which one they are. Remember that you do a test cross, which is crossing it with a homozygous recessive. And so then if they get any little r kids, that can tell you that the parent is big R, little r. So when a human shows the dominant trait, you don't know if they're big R, little r. We for sure would not do a test cross with a human, all right, to see. So that's what that means. Breeding experiments are unacceptable. So what we would do is look at people in their family who are already present to see if they give us any clue as to what the second gene of that dominant individual would be. So, so what we do is we make what's called a pedigree, or a family tree. Pedigree or family tree that describes the relationships of parents and children across generations. And so this can sometimes, not always, give you information about the individual. So inheritance patterns of particular traits can be traced and described using pedigrees. Whoops. And so this is an example of a pedigree. So we're going to look at, so this particular pedigree is looking at a dominant trait of widow's peak. Widow's peak is, if you pull your hair back, if your hair comes to your hairline, comes down to a point right here, that's a widow's peak. If it's straight across, you have no widow's peak. Having a widow's peak is a dominant trait. So therefore, people who have a widow's peak, they use W's here, which I'm not a big fan of, but people who have a widow's peak, what could their genotype be? Big W, or big W, little w, because it's a dominant trait having a widow's peak. So therefore, the people who don't have a widow's peak, we know for sure what their genes are, because that's a recessive trait, which means that they have 2 little w's, right? And so if we want to look and trace this through a family, we make a pedigree. Now there's 2 things on here. There's the square and the circle on here. One is male and one is female. Do you guys know what they are? What's the difference? Male is square. All right, male is square. The universal symbol for male is a circle with the arrow going up to the upper right. I'm going to introduce that to you now because as we get into chapter 15, we're going to do that more. Female is circle, and the universal symbol for female is a circle with kind of a little cross coming down. All right, then that's one thing that you need to know. And then when we look at this, so we have a male and a female. This line here is called the mating line. When you draw pedigrees, it doesn't necessarily have to be humans. They do this for dogs, breeding dogs and horses and things like that. So it's a mating line. We have a line drawn from that and a horizontal line with however many kids they have coming down from this. So these people have 4 kids. Then this couple has 2 kids and their son and daughter married, and they have 2 kids. So notice here, everybody in the same generation is in one line. So everybody in the, and so it's organized that way. But another piece of information on here is that some people are shaded. So the people who are shaded, what does that mean, do you think? They have whatever trait you're trying to trace through the family. Those are the people who are affected with the trait. So the shaded individuals are affected with the trait. Now, sometimes the trait can be dominant, like this example. So this example, the trait we're tracing through is widow's peak. Widow's peak is a dominant trait. So therefore, everybody who doesn't have a widow's peak or is not shaded in, those are the people we know their genotypes for sure, because they have to have the 2 little W's, because to not have a widow's peak, that's recessive. You have to have 2 recessive genes. Which means then that every person who's shaded in has to have at least 1 big W. Let me ask you this. Notice here, they said that he has a big W, which makes sense because he's shaded in, which means he has a widow's peak. But they also put that he has a little w. Can somebody raise their hand and tell me what proof given in the pedigree tells you without a doubt that that second gene for that male is a little w? How do we know without a doubt, what's the proof in this pedigree that that second gene is a little w? Next.
Because the F1 generation has 2 that are not shaded, which means that one of the traits from the male has to be recessive.
That's right. If we look at his kids, he has two kids that are little w, little w. Where do those kids get their genes from? One from mom, one from dad. So dad had to have a little w in order to have a kid like that. If he was big W, all their kids would have a big W, right, and be shaded. All right, so we know him for sure. So this is what I mean by we don't do test crosses, but we look at people in the family that are already present. Who is pertinent to figure out somebody's genotype? You either look a generation up at their parents or a generation down if they have kids. All right, and see if there's any information that tells you what they are. So she's big W, little w, because she had a little w kid. So each parent had to have a little w. Let me ask you this. This woman right here, they wrote that she could be big W, or big W, little w. Can somebody tell me why we can't tell for sure? Why don't I know for sure that she's big W, little w?
Because we have no offspring from her to determine whether she tested or not.
Okay, yep, we don't have any offspring here to help us. Who do we have? Her parents. Now, why can't I figure out from her parents what she is? Abby?
Both parents are heterozygous.
Both? Okay.
They each have a different gene, so she could be either have both dominant for her parents or a dominant one and a recessive.
That's right. Both of her parents are heterozygous, which these are the possible combinations in their kids. So when these 2 parents have a kid that is dominant, they could have a big W kid and her have widow's peak, or she could be big W, little w, and there's no information on here to tell us one way or the other. So she technically could be either one from those 2 parents. Often when you do this, instead of writing big W or big W, little w, you can put a W slash. That means that we don't know. We know she has a big W because she's shaded in, but we don't know what her second gene is. All right. So sometimes you're not able to tell with that. All right, let's look at the next picture, next pedigree. This is a little bit different. This one is tracing another trait through the family. This family we're looking at a recessive trait. This is different. So remember, the shaded people in here have the trait that you're looking at. What's the trait that we're looking at? It's attached earlobes. So when you look at your earlobes, some people, when they come down, their earlobes go straight, attach right to the side of their head. Some people, the skin kind of comes up a little bit, so it kind of looks like you have a little flat, all right, hanging a little bit, so that's the free. So we're looking at attached earlobes. That's the trait that we're tracing through. That's a recessive trait, which means everybody shaded in has the trait we're looking at, which is attached earlobes. So everybody shaded in this case, is little f, using the f's here. Because so when we look at this now, the people shaded in, this is different than here where the people shaded in had the dominant trait. Here the people shaded in have the recessive trait. And so everybody not shaded in has a big F. And so we can do the same thing as we did before. So these people are little f, little f. We know that their parents have to be big F, little f because they're not shaded in. So they have to have a big F. And then they have to have a little f, each one of them, to have the little f kid. So we do the same thing. So when looking at a pedigree, to understand the people shaded in, you have to know whether or not the people shaded in are showing a dominant trait or a recessive trait in order to assign the genotypes properly, all right? And so on. So let me give you an example of this. So over in the margin here on your thing, I'm gonna draw a pedigree here. So let's draw a pedigree. Let's say these two people. So go ahead and draw it because I'm gonna have you fill in the genotype. So we have this male shaded in. Let's say that they have 3 kids. They have a girl, then a boy, and then another girl. This girl gets married and has a little girl, and only 1 kid. And this girl gets married and has a boy, and I'm gonna shade him in. All right, and let's say this family, so that's the family. Let's say the trait we're tracing through is cystic fibrosis, which is a recessive genetic disorder. So what I'd like you to do is, with that piece of information, I'd like you to, for every person next to them, try and write what their genotypes are. So we're tracing the recessive trait of cystic fibrosis through the family. All right, let's go over this. So this is a recessive disorder, so the people shaded in show this recessive disease. So therefore, everybody shaded in this case is, I'll use R's, little r, little r, whatever letter you use. So everybody not shaded in does not have the disease, which means that everybody not shaded in has at least one big R. We know that for sure. So this is what we know for sure. So let's see, as we go through the people, if we can tell what the second gene is. So this particular person right here, this starting at the top, this girl here. So remember, to find the second gene, you either look at the person's parents, which are not included on this, or the kids. So she has 1, 2, 3 kids. Common mistake, don't include this male as one of her kids. All right, he married her daughter. So she has these 3 kids right here. All of them are a big R, and that's all we know. Does that give us any clue what her second gene is?
Say it's big R, because if it was big R, then the probability would be half and half to have a recessive.
Yeah, like so this is what you're saying here. So this is what we're looking at. The dad is little r, and is she big R, little r. So if she's big R, whoops, all the kids would be big R, little r. And could that be the case here?
Yeah.
These 3, all big R, little r. If she was big R, little r, this is the chance every time she has a kid. Every time they have a kid, they have a 50% chance of being a big R, little r kid, 50% chance of being a little r kid. Could these 2 parents, have these 3 kids? They could, yeah. So therefore, we don't know for sure based upon this information. It's like flipping a coin. If you flip the coin 3 times, could you get heads all 3 times? Absolutely, you could. All right, and so it's the same odds here. So therefore, while it may be more likely that this is the case, doesn't prove. There's no proof in this that she is going to be big R, little r, or big R. So therefore, we can't really tell what her second gene is. Let's look at this male here. Can we tell what his second gene is? Absolutely. We look at his kids. His mom and dad are around here, but his kid, he has a little r kid. Where did that kid get their genes from? 1 from dad, and that tells us then 1 from mom, and mom is also little r. This girl, we could look at her kid, but we could also, there's another piece of information, we could also look at her dad. Her dad had little r, which means that she had to get that little r from her dad too. So for her, there's a generation up and a generation down. Both of those are helpful. So he doesn't have any kids, but you look at his dad had little r, so that dad had to give all 3 of his kids. So therefore she would also have to have a little r from that dad. All right, how about him? So she's a big R, little r. We know that for sure. How about him? Can we tell him? No, we can't really tell what he is. He could be big R, and have a big R kid. Or he could be a big R, little r, and have a big R kid. And so we don't know what his gene is. And do we know what hers is? No, we don't know hers for sure either. So she's a question mark there as well. All right, and so what you can do is geneticists will, people who maybe have a genetic disease that runs in their family, they might have a real concern. They know that they don't have the genetic disease, but how do I know that I'm not going to pass it on to my kids and have a kid with the genetic disease? So therefore, doing pedigrees like this sometimes can help in figuring out if somebody is what we call a carrier or big R, little r. They don't have the disease, but can pass it on to their kids. So for instance, she would know, oh, I'm a big R, little r. I have this gene. Her concern might be, okay, what if I have another kid? What if my husband is also a carrier? What's my chances of having a kid with this genetic disease? And so therefore, that might be a real concern. So usually, when we do pedigrees, we're not usually tracing things like attached earlobes through the family. It's usually a genetic disease. There's a reason why we want to know if somebody is big R, little r, or little r, little r. So pedigrees can be used to make predictions about future offspring, and therefore parents can make informed decisions about having children if they know certain things about what the diseases that they're carrying. And we can use some of the rules for probability to figure out, the different chances of kids being born to the family having genetic diseases. And so the last little bit of this looks at some recessive and dominant genetic diseases. So recessively inherited disorders they're inherited in a recessive manner. They show up only in people who are homozygous for the allele, so the people that are little r. So we know their genotypes for sure. It's usually the people who are trying to do all the analysis to see if they're carriers. Carriers, as I said, they're big R, little r. they don't have the disease themselves, but they don't know if they're carrying that gene and can pass it on to their kids, and so that's where the real concern is. And so cystic fibrosis is the example I used here. This is a lethal disease, and 1 out of 2,500 people from European descent have it. We're going to see that different genetic diseases have run in certain descents more than others. And so they have a mutation that causes a defective chloride transport protein in the membrane, and therefore causes thick mucus. I talked about that. Remember I talked about in the trachea where the mucus builds up in the little cilia. Do you remember that? Bringing it up and so on. And so therefore their mucus is really, really thick, these people, because of this defective transport channel. And so oftentimes, because their mucus is thick instead of watery, we usually bring up the mucus to the back of our throats and then swallow it. Remember I talked about that? People with cystic fibrosis, the mucus is so thick because of the lack of water in it that it's hard for that to happen. So usually you have to manually, like, you can pound on the person's back, and they have to cough up the mucus and spit it out, and so on. And so this mucus can build up also in the ducts from the pancreas to the small intestine, and they have issues digesting food, all kinds of stuff. Sickle cell disease is another recessive disorder we've talked about. This is more prevalent in the African American community, 1 out of 400 people, and it's based upon just one mutation, one base in the DNA is changed, which changes the amino acid, which changes the structure of the hemoglobin protein. We talked about that. Remember that I showed you a picture of that when we did proteins. The hemoglobin causes the red blood cell to become sickle shape, or almost like a half moon kind of shape, quarter moon kind of shape there. And so that's a recessive disease as well. Because of a lot of genetic diseases being recessive, you can be carriers and not know it. And which is why mating of closely related organisms, so this could be within any organism within the animal species, closely related organisms have a higher likelihood of carrying the same genetic disease. So like for cystic fibrosis for people, if you're related, you have a higher likelihood of both being a carrier for that cystic fibrosis gene than if somebody who's not related to you. There's a lot higher likelihood that that doesn't even run in their family. So people or organisms, dogs, this happens with horses if they are interbred within the same family. What happens is they are carriers of these same recessive diseases, and so when they have kids, they both pass on the little R, and the kids can have all kinds of genetic problems because they're carrying this, the parents are carrying the same genetic diseases. And so certain types of dogs, if they're interbred too much, are known for having certain things wrong with them, and that's because of carrying the same recessive genes. All right, so that's why that's a bad thing to interbreed too much. Some disorders are dominant, and so in humans, chondroplasia is a type of dwarfism. If you're big R, the individual doesn't survive enough to have long enough to have children if they're homozygous dominant. So if they're big R, but they do survive if they're big R, little r. So you can have a person that's big R, little r, and has this chondroplasia, and so you can have two individuals that are big R, little r, have children. Is it possible for them to have a child of normal height? Absolutely. All right, if the two little r's come together. All right, and so for instance, there's that TV show. What is it called?
Game of Thrones.
No, there's a TV show.
It has.
Little People, Big World.
Yeah.
Is that what it is? All right, where it's a live reality TV show of two parents that have some dwarf and some
There's
normal sized
a
height.
couple TV shows that follow.
Oh, is it? But this one particularly shows what I was talking about, normal height versus, yeah. All right. Huntington's disease. Huntington's disease is another dominant disorder in humans that is a degenerative disease, which means the nervous system over time breaks down and gets worse and worse over time. And the thing is, so to have Huntington's disease, you're big R, or big R, little r, right? The thing is that the disease, you don't even know you have it. You have no symptoms of this disease until you are 35 to 40 years of age. Then you start to see symptoms. And so the thing with this then is that usually by 35 to 40 years old, people may have already had children and could have already passed on that gene to their kids before they even know that they have that disease. And so the good thing about Huntington's disease, though, is they now have a genetic test where they can take your genes and test to see if you have it. And then you kind of know if it runs in your family because you can look at your parents and grandparents and see if other people had it. So you know whether or not you have the chance of having it. But the thing is that you usually have to be tested genetically to know. Unless you know that both of your parents don't have it, then you would know. If both of your parents are little r, and they never got it, then you can be reasonably sure.
Is there a possibility, for example, Huntington's disease, when your parents don't have the disease or that trait, for you to just kind of develop it in like a mutation?
What would have to happen is that if they don't have it, either the mom or the dad in this egg or sperm cell, if they had a random mutation in there, then I guess that could have happened. That's real bad luck there. But no, you can't just develop it. It would have had to happen early on in development. So either the mom or dad has so they don't have it, because technically you could not have the disease, and if in your egg cell a mutation or a sperm cell, a mutation can happen, so then you pass on something that you necessarily don't have. You can pass it on to your kids. That's how originally genetic variation enters in a population, is by that random mutation. So it is possible. Okay. But that's what would have to happen. All right. I've already talked a little bit about this, how lots of diseases that are multifactorial have meaning many factors, things that go into its expression. So heart disease I talked about, cancer, have both genetic and environmental components. And so they're still looking at a lot of what's genetic versus what's environment and what plays a role in the expression of these genes. So people can see a genetic counselor, and the genetic counselor can do things like take family history, like making a pedigree to give you information about if you're big R, little r. They can help to determine the odds that your children will have genetic disorders so that parents can make informed decisions about whether or not they even want to have children, depending upon that. There are, for a lot of diseases now, and it keeps growing and growing and growing, the more research that's been being done, where you can get a genetic test to test if you are carriers. As a parent, remember a carrier is a big R, little r. So if you have the dominant gene, you might want to know if you're carrying the recessive disorder that runs in your family so that you can tell if it's going to be passed on to your kids. Then if you already are pregnant, if the woman is pregnant, you can also test the fetus through various ways. In amniocentesis, the liquid that bathes the fetus is removed and tested. And chorionic villus sampling, or CVS, a sample of the placenta is removed and tested. And then there's also ultrasound that actually tests the baby while it's in the uterus. So it's assessed visually. So amniocentesis, let's talk about this. Amniocentesis is, oops, wrong way. What they do, here's the baby. The baby is growing in the mother's uterus. let me explain here. There's what's called an amniotic sac in the uterus that has amniotic fluid that the baby is floating around in. So what they do is in an amniocentesis, you can see here they take a big needle and they puncture through the uterus and into the amniotic sac, and they suck out some of the amniotic fluid from the inside the uterus. And so then they bring it out, so you have the sample of amniotic fluid. Why you want that is because there are cells from the baby. You know how our cells slough off? There's baby cells in there that you can then get the DNA from and do some genetic tests. You can do karyotypes to see if the baby has the right number of chromosomes, and so on and so forth. So that's one way to get information about a baby. You might want to know that ahead of time for some, like if they think there might be a genetic disease, there's certain care that might be needed for right after the baby's born that you can be prepared for, and so on. Chorionic villus sampling, or CVS, is taking a piece of the placenta. So we talked a little bit before about the placenta. So they take a suction tube through the cervix, so they go up through the cervix and suction out part of the placenta and can get cells that way. And then the ultrasound, that last one, I don't have a picture of, but it says, gosh, you can assess the baby visually, and ultrasound uses ultrasound radiation. And it's just a wand that they put on the mother's stomach. They put some gel on it and put it on the mother's stomach. It beams this ultrasound into the womb, and then on the screen, there's a computer screen that shows you a visual picture of what the baby looks like and so on. And they usually do that as a general rule about halfway through a pregnancy. They do measurements and so on. They can tell if the baby's growing properly and so on. And also at that point can see if there's something wrong, where they might then want to then go back and do some of these other tests, all right, for more research. It's very low. Yeah, no, like it's not harmful. I shouldn't use that. Yes.
Don't they start the ultrasounds like the first month of pregnancy?
Well, if it depends on the person, but no, usually not. That's not a typical. Usually, most people.
Seems like late.
No, because they can get measurements so that they want, like even halfway. Really, like when you see the ultrasound picture, like sometimes it's like you don't even know what you're really looking at.
Yeah.
Like, you know, like it's loosely looks like a baby. You know, so the ultrasound technician, like, I mean, when I went for mine, there's no way I could have been able to tell if my kids were a boy or a girl, or like.
Yeah, it's like, well, it's a boy. It's just look. It's like.
Yeah. You have to be trained for it for most people. When they're smaller, they can do it, maybe for high-risk pregnancies and so on, but usually for higher risk, they'll do more ultrasounds after that halfway, but before that, there has to be a reason for it. Usually, it's not normal to do that. So if it is being done, there's something that they think is going on. All right, for that. So like halfway through my pregnancy, I got diagnosed with type gestational diabetes, and so then I started like my last 2 months. I was getting ultrasounds like every week. They wanted to go in and they would do it. Like they had to, because there's issues with the baby with their head and shoulders being too big, like if you don't monitor your diet and so on. So there's issues with the health of the baby and stuff like that. So they just want to make sure everything was okay. So they're usually a reason for it. All right, having multiple. Okay, so all right. And then the last thing, there are certain tests that are done after the baby's born with newborns screening. So certain things are easy to detect at birth for preventative measures. Yeah. It's a good one.
It was like pretty much This allergen. Like one guy like tries to pretend that he's but like working in some big company See, what question are you? Did you finish the final? I'm pretty sure.
It's
It's kind of cool.